Okay, so today we're going to be presenting on ELISA, which means enzyme-linked immunosorbent assay. And my name's Nia Dashal.
My name's Tori.
I'm Nick Vicini.
My name is Shanona. Oh my God.
So what is ELISA? So as Anina said, it's an enzyme-linked immunosorbent assay. So what it does is it detects and measures antibodies in the blood, and it can determine if you have antibodies related to any certain infectious conditions. So the key people involved.
I'm sorry. Okay, go.
So the key people involved in developing this test. Next slide, please. So there are two main people. So they're both Swedish scientists. The first woman is Eva Engdahl, and the other man is Peter Perlman. So they were just the two people involved in developing this test. So here's just a timeline of developing the ELISA test. So in 1960, first it started off with radio immunoassay, and then 1966 led to non-radio immunoassay. In 71 came methods to perform ELISA, and then eventually in 2012 the ultra-sensitive enzyme test was created, which is ELISA.
Now the key science and principles involved in the test, molecules involved, reaction and detection of the reaction. Next slide.
You can't see the next slide?
No.
It doesn't say molecules?
No.
Mine does.
Try going back and then going forward again. See that? There we go. Molecules. Oh.
Could you see it now?
Yeah, I got it. Molecules involved in ELISA include horseradish peroxidase and alkaline phosphate. So they discovered like a new form of horseradish peroxidase called a streptavidin polymeric horseradish peroxidase complex conjugate, which causes a typically 19-fold stronger signal generation in comparison to the traditional monomeric horseradish peroxidase. And they also use alkaline phosphate conjugated antibodies. This is used to combine with a chromogenic substrate solution, and this allows them to detect the immune response in these enzymes. Next slide.
Can you see it?
Yeah, there we go. Reaction and detections. And so the most prevalent detection methodologies for ELISA are colorimetric, colorimetric, fluorescent, and chemiluminescent. During the detection step of the protocol, a substrate for these enzymes is added. They dilute the detection antibody in sample dilution buffer. They may add a second dilution antibody. Then they add a substrate solution. They allow it to incubate as the color develops, and then they add a stop solution to stop the reaction when the color is sufficiently developed. And then, of course, you measure the absorption via a plate reader. Next slide.
Okay, types of, can you see it?
Yeah.
So there are three types of ELISA: colorimetric, fluorescent, and chemiluminescent. So the first one is the most commonly used type of detection for ELISA assays. The amount of color in each is well read by a spectral photometer, and the samples are compared relative to one another with use of a standard curve derived from a known analytic concentration. And then the next one is fluorescent, and this kind of ELISA is useful for measuring immune responses, and also if a molecule is too small for a sandwich assay, it provides a useful tool for quantitative measures. And the last one is that allows for the advantages of the detection which are typically higher in dynamic range and lower background signal, leading to increased sensitivity. And this signal is not as stable as the other two detections types and must be read within a short period of time, generating the signal. So methods, there are 4 types of methods. So the first method is direct ELISA, which involves an enzyme conjugated to an antibody that binds to an antigen on a surface. The second one is indirect ELISA, which is similar to direct ELISA, but the antibody is not conjugated, and a second conjugated antibody is then used to detect the bound antibody. The next one is sandwich ELISA, and the antigen is recognized by 2 antibodies, making a complex like sandwich, and one antibody is then used for capture and one is used for detection, and then the detection can be direct or indirect. The last one is competitive sus inhibition ELISA, which is similar to direct ELISA, but the quantitation is performed by competing or inhibiting the antibody binding with the measured amount of antigen.
Okay, so where and why was it first used? And like what were the circumstances at this time? And the ELISA was designed by 2 Swedish scientists as toy before, and it was used to identify whether or not someone was HIV positive. It was made in 1971, and during the 1970s and like starting in the 1960s, there was a huge outbreak of HIV, specifically among the LGBT community and among drug users.
Okay, so is it used at the present, and then how is it used currently? So these are just a few diseases that ELISA test may be used for to diagnose. So these include HIV, which causes AIDS, Lyme disease, pernicious anemia, Rocky Mountain spotted fever, rotavirus, squamous cell carcinoma, and syphilis. So ELISA is used in the present day, and it's often used as a screening tool before they order more in-depth tests. So these are the steps of use for the ELISA. So first, a blood sample is taken and it's sent to the lab, and then the technician adds the sample to a petri dish containing the specific antigen related to the condition that you're being tested for. And then if the blood has antibodies to the antigen, the two will bind together, and you may have the condition if the contents change color in the dish.
Okay, for the details about the companies and organizations making the test, we're going to talk about location management and valuations. So the primary organization producing a test was founded in 1990 and is called Molecular Innovations. This is like the main producer of the test, but ELISA is partnered with over 100 organizations, and just to name a few, there's Abexa, BioAssay, eBioscience, Immunospect, Cometa, and Vector. These agencies use the ELISA test in order to do their own like molecular sciences, and Molecular Innovations, they make a revenue of about $10 million per year. So since they've been open for 30 years, you can estimate that their net worth is about $300 million. Some of these other companies as well, the organizational partners, they are worth over $1 billion.
Can this be used for COVID-19 testing? Why? Coronavirus that causes COVID-19. Researchers say that ELISA works like antibody tests for other viruses such as hepatitis B, and will show whether your immune system, the antibodies defense against germs, made contact with SARS-CoV-2 even months before. The test could help scientists fight the pandemic in several ways. It can give researchers a more accurate measure of how how many people had the coronavirus. It would also help let healthcare workers who were ill with COVID-19 symptoms but were never tested for the disease return to work. It will it might also help with an experimental treatment for COVID-19 called convalescent plasma. And the therapy, doctors collect blood from people who have recovered from the disease, and researchers hope antibodies in the blood can treat people with COVID-19. The ELISA test could help identify people with antibodies. These are references. Thanks. Any questions?
Thank you, guys.
Thank you.
Yay.